Okay, we're going to talk about new drug treatments for cystic fibrosis. So cystic fibrosis, if you are familiar with it, it's an inherited disease, which people who suffer from it suffer from thick mucus in the lungs that causes repeated chest infections, which eventually leads to damage of the lungs and shortened life expectancy. Right, now it is a genetic disease, and it is due to a mutation in this protein here, which is called the Cystic Fibrosis Transmembrane Conductance Regulator. It's a membrane protein, and it acts as a chloride channel. So it lets chloride ions pass out of cells down the concentration gradient out of the cytoplasm into the extracellular space. Right, let's have a look at the gene there. So we've got the N-terminus there, right? And then you get this blue domain, which is, that is a transmembrane domain. Then this red domain here, that actually binds ATP, nucleotide binding site there. Then there's an R domain, all of the same peptide chain, R domain, and that's got multiple sites where it can be phosphorylated there. And controls the protein. The green is another transmembrane domain, and the blue and the green together form the channel through which the chloride ions pass. And then finally, before they reach the C terminal, we have this orange domain here, and ATP can bind to that as well. Okay, so that is the protein. Normally, it allows chloride ions to pass out of cells. Mutations in cystic fibrosis mean that this doesn't work properly, as we'll see in a minute, and we'll see how that causes the thick mucus as well. But just have a word about this conductance regulator, the CFTR. It's part of a much larger family of proteins, and they all bind ATP. They're called the ATP binding cassette, ABC. There's a whole lot of proteins. They all come from the same evolutionary origin, so there's a lot of sequence homology and so forth between them. Most of them, like the sodium potassium pump here, so you should be familiar with that from nerve impulse stuff. It's present in all cells, of course. Right. Most of them actually pump ions against the concentration gradient, unlike the CFTR. So what happens? This is a typical one of these proteins. Pumps is you've got the blue, which is you've got a transmembrane domain there, and it binds ATP. ATP gets hydrolyzed here, so we've got ADP in organic phosphate, and the energy from that is used to change the conformation of the protein. So you can see I've changed the way the two blue bits are interacting with each other, and as a consequence of that, you can see here that we've got 3 sodium ions binding on the inside and 2 potassium ions binding on the outside. When this changes shape, those ions move across. So here the sodiums move out and the potassiums move in to the cell. Again, that is against the concentration gradient. So essentially the energy to provide for that, it comes from the hydrolysis of ATP, but indirectly the ATP is used to change the conformation of the protein into a more energetically less favorable state, and that's how it is achieved. Okay, so just to show that there's not actually it's a member of quite a large family of proteins, but there's a bit of a difference here. Is, as we say, the CFTR doesn't pump ions out. Chloride ions, and call them here, they just pass through passively down the concentration, down the electrochemical gradient outside of the cell. Okay, so we need to talk a bit about this. If you look in any of the articles about this, we talk about how the CFTR is gated. Okay, so you probably come across voltage gated channels. You get a change in intracellular membrane potential, and that will open sodium channels, or ligand gated, acetylcholine, that's a good example of one, where acetylcholine binds to it, changes the shape and switches essentially opens or closes the channel. Now the CFTR is pretty complicated how that is gated, and in normal in the so-called wild type protein, the normal protein, it seems to have a high probability of being open most of the time. Okay, this is being open. Okay, right, and in order for it to be open, it does seem to need ATP to be bound. You're not really going to need ATP hydrolysis here because the chloride ions are moving down the concentration gradient. However, some ATP does get hydrolyzed. That's been established. Also, this R unit here, if this is this phosphorylation status of this. So there are multiple sites that can be phosphorylated on that, serine residues, by protein kinase A, which you've probably heard of. That's involved in glucagon. How glucagon exerts its effect. It's cyclic AMP dependent. So that can be phosphorylated there. And depending on how much it is phosphorylated will mean it's open. It's either going to have a high probability of being open or a lower probability of being open. But as I say, essentially, in the normal protein, it's open most of the time. Cystic fibrosis, we can see certain mutations mean that the probability of that channel being open is drastically reduced, and so the chloride ions can't get out the cell. Right, so let's have a look at why chloride ions, why should that be? Why? Right, so I'm just going to get rid of that. Right, just drag this over. Right, so this is how has this effect. Okay, so I've drawn some lung epithelial cells. So here we're going to have, this is the extracellular, this is the airway here. And here's the, inside the body on that side. Right, so we can see here that the chloride ions in green, they are allowed to pass through the CFTR protein, which I've done in red. They pass through there. The chloride ions will pass out down the concentration gradient. Of course, they're negatively charged, so that will change the membrane potential a bit, which means that the sodium ions, which I've done in blue, tend to follow passively right through channels because if a negative ion goes one way, a positive ion is going to go that way, the same way, to stop changing the membrane potential. Right, now because you effectively there, you are allowing sodium chloride to exit the cell, that is going to lower the water potential here. So that means water is going to flow out of the epithelial cells to the outside. Now here I've written here there's mucus, and that will make this mucus watery. And the mucus needs to be fairly watery. In cystic fibrosis, you don't get the chloride ions going out, so you don't get the sodium ions going out, so the water doesn't go out, so the mucus is much thicker. And the little cilia, which moves the mucus about, can't do that effectively. You get repeated chest infections. Each time you get a chest infection, that causes lung damage, which causes makes the lifespan shorter. Also, the CFTR protein is quite important in the pancreas. So in the pancreatic secretions, without this chloride coming through and the water coming through, they're too thick and they get blocked, so there's poor digestion and so poor absorption of food, which also contributes to the disease state. Right, so let's talk about mutations which actually cause, get rid of that, I'll copy that. Let's talk about the mutations which cause that. Right. Okay, so right here, just clean this up a bit. There are quite a lot of different mutations in cystic fibrosis. Now I'll talk about one of the most common ones here, which is called it's called delta 508, and what it's called delta 508 because you've actually got a deletion there of a whole amino acid. There should be a phenylalanine at 508, and the whole amino acid is missing. Right, and that actually, that deletion is in that part of the protein. So it's not the transmembrane bit, it's the bit where the ATP binds. So that's where you have a deletion there. That will cause this protein to fold incorrectly. Right, that's a very common mutation. We'll talk about that in a bit more detail. There are also other deletions. Oh, it's not deletion. Sorry, there's substitution. So there are quite a lot of point mutations. And that's where you get one base change. So you get one single amino acid gets replaced. This is exchanged for another one in the sequence of the protein. That may happen and may not cause any ill effects at all, but there's a whole range of mutations which have been identified, point mutations, which lead to a substitution, and it leads to reduced conductance. Now, what that means, reduced chloride ion permeability. So the chloride ions can't get through as easily as they should. Either they don't, the probability of that channel being open is 0. They can't get out at all, or it's very much reduced, so you don't get enough chloride ion going through there. So they're the two main kind of different mutations there. Let's look about this delta 508 mutation first of all, because I say that affects 80% of the people who suffer from cystic fibrosis have that mutation. Right. Oh, by the way, they've got delta 508 there. So, these are the ones. So that 551, that's a very common substitution. So you swap a G. It's position 551 in the protein, and you swap G, which is glycine, for D, which is a single amino acid code for aspartic acid. Or this one is another one, 1303, position 1303, and in that case, you're swapping an asparagine, which is N, for K, which stands for lysine. So these are two very common mutations here. So let's look at the delta 508 one first of all. Right, what happens with the deletion is that the CFTR protein doesn't fold properly. So what should happen is the nucleus, of course, makes the mRNA transcript. The protein is synthesized. It's a membrane protein, so it's synthesized on ribosomes in the rougher endoplasmic reticulum. I've drawn the CFTR protein as little red dots there. They are then trafficked to the golgi apparatus. The golgi apparatus puts them into vesicles, and the vesicles then fuse with the cell membrane here, and so that's how the CFTR protein gets the cell membrane. That's where it should be. Now, in the case of the deletion, delta 508, sometimes delta F508, F stands for phenylalanine, by the way. Right, so what happens is the protein doesn't fold, or it has a reduced probability of folding correctly. You should really say some of it folds okay, but a lot of it doesn't. So what happens when it gets as far as the Golgi recognizes it's gone wrong, and it gets marked for degradation. Okay, so it never gets to the vesicles, it never gets to the cell membrane. So people with that mutation have much reduced levels of the CFTR there, and when it is there, of course, it's mutated as well. We'll talk about that later. So the main problem with this is that it doesn't get to the membrane. So what can be done about that? Well, it was realized, it's a folding problem. It's a protein folding problem. So normally, as proteins are translated at the ribosome, they fold. Some proteins will fold okay by themselves. Some of the proteins need something called a chaperone protein, and there's a lot of these proteins, but sometimes a lot of the heat shock proteins, if you've ever heard of those. Chaperone proteins are proteins which kind of bind to a protein as it's been translated and makes it fold in the correct way. And it seems that the CFTR protein does need a chaperone protein. So the approach was to look for small molecules. Okay, so the small molecules, which when you add it to this, so you're going to culture some cells with the delta F508 mutation in them, and you're going to add tens of thousands of different small molecules, tens of thousands, lots of them. It's called a high throughput throughput screening, and you screen those compounds to see if any of them, by chance, can improve the folding of that protein. And the way they did this high throughput screening was you look for the CFTR on the cell surface with monoclonal antibodies with a fluorescent tag on them. So you're looking to see, is the protein getting to the cell membrane in sufficient quantities, or is it ending up getting degraded in the cytoplasm? And they did tens of thousands of molecules here. Now, they came up with about 10 molecules out which worked okay. Some of them are very toxic, so they couldn't be used as a drug. But if we have a look at these, sorry about that. Right, there are, well, we'll look at these two first of all. This one, which they called tezacaftor, and this one called lumacaftor. They're pretty small. There's the chemical formula you can see. They're pretty small, they're not like proteins, huge tens of thousands, There's an MR of about 500 or something, those things. And they, most of the compounds didn't have any chaperone effect at all, but these did. These seemed to help the folding of the protein when it had the delta 508 mutation. Okay, and you can see there is quite a few similarities going on there. Like they both got this this funny little cyclopropane ring, and then you've got it's virtually identical in this bit here. So that's obviously pretty important in the binding of the protein, and then it's a little bit different the other end there. So they have actually been used. They're used as drugs, and they help the delta F508 mutated protein not to be degraded in the Golgi body, but to actually get to the cell membrane where it can work and let the chloride ions out. More recently, there's this one which they've called Alexacaftor. It's the same company who've done all of these. That's second generation. You can see it bears no obvious resemblance to the other two at all from the formula of it, and that improves the trafficking, the folding of the protein, and the trafficking to the membrane. Now, that is not the only drugs they have done. They've also did again one of these high throughput screening, and this time they're looking for this one. I have a Kafta. It's called a potentiator. And what the potentiators do is they bind to the CFTR, and somehow they increase the probability of the channel being open. And as I said before, the actual control of the channel being open is not very well understood at the moment. They do realize that you need ATP to be bound to the ATP binding sites, and you also need some phosphorylation of biprotein kinase C of some sites in the CFTR protein. Again, it's not really known how this potentiator molecule works, but it obviously does bind to the CFTR protein and increase the probability of being open. Again, they identified it through one of these high throughput screening. So they got a load of cells with mutated CFTR, cell line with mutated CFTR, and this time they looked at the ability. They were not looking at with the chaperones. They're just saying, oh, do they get to the surface? We're saying, do they work better? So they looked at the ability to pump iodide ions. Now, iodide ions has also got the same charge as chloride ions. They are a bit bigger, but the CFTR molecule can act as an iodide channel. And what they did was they got a cell with iodide in it, and outside of the cell, they had a fluorescent molecule which will react with iodide. So if the iodide's getting out, it reacts with the fluorescent molecule and stops it being fluorescent. It's quite a simple test, and it needs to be quite simple because they've got to screen tens of thousands of compounds in the hope that they find one that will work okay. So it's kind of like, it's not a drug design, a rational design. It's just get lots and lots of different compounds and see if hopefully one of them will work. And they did find ivacaftor that does work. Now, new treatments of for cystic fibrosis, most of them, well, these chaperones, these are only going to work on the delta 508 mutations, because some of the other things, they get to the other mutations get to the cell surface, no problem at all. It's just that they don't really conduct chloride ions. But this delta 508, the major problem is it's not getting to the membrane. So therefore that. But in combination with them, they also add some of this ivacaftor, because even the delta 508, even when it does get to the cell membrane, it's not brilliant at letting chloride ions through. So if you can then add a molecule which will bind to the CFTR and increase the probability of it letting chloride ions out, that's going to work even better. So most new therapies, as I say, there's 4 different drugs there. There's various combinations of maybe 2 of them or 3 of them. But generally speaking, you're going to need a chaperone molecule and a potentiator molecule, and they're taken together in 1 tablet daily. The main problem with this treatment is incredibly expensive. It's about £300,000 per patient per year, and that is kind of outside of the health budget. It's very difficult to justify spending that much money on one person, even though it's a life-saving drug for them. They're going to need that for a whole life, for a whole lifetime. However, the prices do seem to be coming down. I don't know the reason. I don't know the exact reason why the prices were so high in the first place, but it does seem a promising treatment. Another interesting thing about cystic fibrosis is this concept of an orphan drug. Right, so orphan drugs are drugs which treat diseases that not many people suffer from. So there's not a great deal amount of money to be made for them. So the dose per dose basis cost is going to be very high. Cystic fibrosis is a fairly common disease, but it's nowhere near as common as something like asthma or something, or where you can sell drugs to millions of people. This is a much smaller market, and so in the past, pharmaceutical companies have been very reluctant to develop drugs for these sort of diseases. All of these drugs were developed in a combination between a charity, a cystic fibrosis charity in the United States, and a drug company. It was a new approach to hopefully develop some of these drugs for orphan conditions where there's just not enough people suffering from the disease to make it economically viable.